So that covers the first part of urine production, glomerular filtration. Right? The next two parts are tubular reabsorption and tubular secretion. Okay? And the most physiology really occurs with tubular reabsorption. Okay? A lot of material gets pushed out through the glomerular filtration, through the filter, more than what we want to get rid of. And so reabsorption is a reclaiming process. Takes back the material through a selective transport. Okay? From taking material from the tubule fluid and putting it back in the blood where it came from. Okay? When this happens, the filtrate composition is altered. Okay? At first, the filtrate is very close to plasma. Okay? In constituency. All right? It's close to plasma without the proteins, but now it's a little bit different. Okay? And about 60 to 90% of the filtrate is actually going to be reabsorbed. All right? Remember, we produce about 180 liters of filtrate, but we only actually produce about 3.5 liters of urine a day. Okay? And most of this occurs in the proximal convoluted tubule. All right? Most of this in the PCT, but not exclusively. Okay? Substances that are reabsorbed can be classified into three groups. Things that are freely reabsorbed, right? They happen, we need those, they come back. There are things that are reabsorbed with some regulation, okay? And then there are things that are not reabsorbed, and therefore they're eliminated as waste. Okay, some three different categories. All right? So you can kind of think about the first category as completely reabsorbed. This is stuff we need, right? That you need in your body. Okay? So how does this happen? All right, so here's the peritubular capillary. This is really an exchange between the peritubular capillaries and the tubule. All right, and it is regulated by the epithelium of the tubule. So here you have simple cuboidal epithelium. And transport can occur in two different areas. All right, we have to go through the cells or in between the cells. All right, so between the cells is paracellular transport, right, between epithelial cells. And these epithelial cells have connections between them, tight junctions, that will not necessarily allow this to occur only for certain substances. And then transcellular, there's a protein or a channel or a carrier that will move the material from the lumen of the tubule through the cell and then out of the other side. All right, so transcellular, it has to go through the cell. Okay, and then, so here's tubular reabsorption. Substances move from the tubule into the blood, and tubular secretion goes from the blood to the tubule. It's reabsorption in reverse. Okay? Questions on the basics of it? Okay? And these are transport proteins, channels, porters, carriers, pumps are very important in this process. Okay? Some of these processes of movement of materials is active. Some of it is passive. Okay? But what you end up with is what's called a transport maximum. Okay? Because this is regulated by proteins and the movement of proteins, okay, and the ability of proteins to move materials from one side to another, there's a certain point where this kind of substance can't go any faster, or so much can be moved. Okay? So it's transport maximum is the maximum rate of a substance that can be reabsorbed or secreted across the tubule epithelium in a period of time. Okay? What does that mean? Let's use in terms of glucose. Okay? Really all of this depends on the number of transport proteins present on the membrane. The more proteins, more transport proteins, the more material can be moved across. Okay? So if I want to move glucose from the blood into the urine, or from the urine back into the blood to reabsorb it, I need to have glucose transporters present on those cells. And if I have more glucose transporters, I can move glucose back into the blood quicker than if I have less. Okay? So the abundance of a transport protein is directly related to how fast the substance it's transporting is moved. Understand? Okay? And then there's a renal threshold. Okay? A max plasma concentration of a substance that can be transported in the blood without appearing in the urine. Think about that. There's a renal threshold. Okay? It's the maximum amount of a substance that can be in the blood before it ends up in the urine. Okay? Glucose is a great example. Okay? Normally, glucose is not found in the urine. Okay? Normally, it is reabsorbed. It is a nutrient. It is one of those molecules that is freely reabsorbed, that first category. Okay? But if I have a high glucose diet and a high sugar diet, if all I do is drink Mountain Dew all day long, Lily, okay, all day long, what happens to my blood sugar? It goes up and it stays up, right? And if I have an excess amount of blood glucose, I reach this renal threshold where it's reabsorbed, and now it's going to be passed in the urine. Okay? I mean, it's that simple. I reach a maximum where all of the glucose transporters are doing their job, and there's still excess there. It needs to go somewhere. Okay? It doesn't get back out, doesn't get reabsorbed. Okay? So here's glucose reabsorption. Okay? Low sodium, high glucose. So here's your tubular fluid at the bottom, blood up top. Okay? In the normal situation, what occurs is that blood glucose is reabsorbed. It's transported in a concentration gradient back into the blood. Right? Pretty straightforward? Yes? But if this is too high, okay, that means blood going into the afferent arteriole is going to push glucose in here. This is going to be high. Okay? This still occurs. This high glucose movement still occurs. But eventually what's going to end up happening is that right here is going to end up causing a little bit of backlog. Okay? Your concentration gradient isn't established for glucose, isn't established. So this movement is disrupted. It ends up with an excess amount of glucose in the urine. In other words, glucose is not getting reabsorbed, right? And you end up with glucouria, or glucoseuria. Glucoseuria, there's an S in there. Okay, glucoseuria. It's indicative of the first sign of diabetes. Okay? Questions? So glucose is a great example of those substances that are 100% reabsorbed, freely reabsorbed. Nutrients, glucose, amino acids, proteins. Right? Protein should be reabsorbed as well. Okay? So that's pretty straightforward, those substances that are 100% or freely reabsorbed. Okay? Goes by those transporters, and only if there's an abundance of them in the blood or would they not be reabsorbed. Would you find some type of those substances present in the urine? Now, substances that are reabsorbed with some regulation include sodium, water, potassium, calcium, bicarbonate, hydrogen ions. All right? Because they also regulate other stuff. Okay? Think about, I just said water follows salt and chlorine, right? Water follows sodium. So when you reabsorb sodium, what's coming with it? Water. Okay? If I reabsorb hydrogen ions, they have to go somewhere, right? And that could lead to a change in blood pH. If I don't reabsorb bicarbonate ions, I'm getting rid of them, and they're important in regulating blood pH. Okay? So there's this other stuff that are involved with some of these other materials. If glucose doesn't get reabsorbed, does anything happen? It ends up in the urine, right, which then causes kidney problems, but is the blood pH going to change? No. Is the amount of water that's going to end up in the body going to change? Not really. Okay? So this is why these are regulated substances more than, say, nutrients that need to be freely reabsorbed. Okay? So most of the sodium that gets out in the filtrate is reabsorbed, about 98 to 100%, okay, in a normal diet. Okay? Most of this occurs in the proximal convoluted tubule. Okay? And the concentration is relatively low inside the tubule itself, right? But it's high within the tubule lumen and the interstitial fluid. In order for this process to continue, right, we have to always maintain a concentration gradient. It's going to go from tubule to cell, and then from lumen to cell, from cell to interstitial fluid. Okay? So it moves down its gradient across the luminal membrane into the tubular cell, and then a sodium-potassium pump will help pump it into the interstitial fluid. Okay? And this is actually regulated by a couple different hormones, okay, including aldosterone and ADH. Where am I? Okay, so sodium reabsorption, regulated by hormones, aldosterone, and ADH. All right, so here's aldosterone, and this picture is actually taken from chapter 25. I think this is 25.4 or something like that. All right, so this is chapter 25 is fluids and electrolyte balance. They're not separate. Okay, every anatomy book that you will read will separate them from one chapter on the urinary system, another chapter on fluids, electrolytes, etc. But they're really not. They're directly related. Okay, kidney function and what it does is do this. All right, so here's the stimulus: a decreased amount of sodium in blood. So we want to have a relative amount of sodium, right? And so in order for this reabsorption to occur, if sodium's low, we need to reabsorb more sodium. Now we're already about 98%. Okay? So aldosterone is released in response to angiotensin II. Okay? Response to the stimuli, the cortex releases aldosterone in the blood. So here's aldosterone. What happens? It affects the kidney. Okay? The kidney is going to increase potassium secretion. Right? That's dumping of potassium into the excreted part, into the tubular fluid. Okay? But what it does is increases sodium and water reabsorption into the blood. So aldosterone increases sodium reabsorption. And why do I include water with that? Because water follows sodium. Okay? Because now if I bring in more water, okay, blood plasma now, sodium's going to go up, okay? But the blood volume and blood pressure is going to be maintained because I'm going to reabsorb more water. What happens to urine? If I'm reabsorbing more water, what happens to the amount of urine I'm producing? Mary? It decreases. Okay? So all of this is related. All right? So this is one way of affecting not only sodium concentrations, but also the amount of urine that's produced. Okay? By aldosterone. Aldosterone reduces urine production by increasing sodium absorption, right, and water absorption. Okay? We already talked about HO natriuretic peptide. Okay? So now, HO natriuretic peptide is released by the heart in response to increase stretch, increase blood volume. Increased blood volume means there's too much water, so we're going to get rid of sodium. What do you think? Are we going to increase or decrease reabsorption of sodium in order to get rid of it? Decrease. Okay? So atrial responds to stimuli, release AMP into the blood. What it goes down to, it goes to the systemic blood vessels, vasodilation, right? Allows more blood to go through the kidney. But at the kidney, because of vasodilation, we have an increased GFR, more blood running through the glomerulus, more filtrate being produced. More filtrates produced, okay? It helps to decrease blood volume. And in the nephron, you get increased loss of sodium and water in the urine, which will counteract the stimulus of an increased blood volume and blood pressure. Okay? So, aldosterone and atrial natriuretic peptide work, right? They help regulate the amount of sodium that is absorbed. Okay? So if sodium is regulated reabsorption, so does water. Now, those two we just talked about, ANP and aldosterone, they also affect water reabsorption, right? Yes, they do. All right, but that's based upon the amount of sodium that's produced. All right, so water is also reabsorbed. Water moves through the channels on cells, aquaporins. Water movement is a passive process, right? So it's reabsorbed by both paracellular and transcellular processes, right? Transporter proteins, aquaporins. All right, about 65% of it is reabsorbed in the proximal convoluted tubule, okay, before it gets to the loop of Henle. All right, and water follows sodium by osmosis. You're going to move sodium from one side to another. You're changing the concentration of water on one side, and water will follow. Okay? This is what we call obligatory water reabsorption through reabsorbing through the movement of sodium. Simply put, water follows salt, or water follows sodium where it goes. Okay? In the nephron loop, 10% of the water is filtered, filtered water is reabsorbed, and then this also occurs in the distal convoluted tubule, collecting tubes and ducts, right? And that's controlled by both aldosterone, which you just talked about, and a third hormone we'll talk about in a minute, ADH. ADH, antidiuretic hormone, right? You want to know what it does? You can think of it as this: anti-P hormone, or anti-urine hormone. So what is it going to do? Is it going to increase urine production or decrease urine production? Olivia? Decrease. All right? Antidiuretic hormone. Okay? Aldosterone, okay, increases sodium potassium pumps and sodium channels, right? Thus increasing water reabsorption. So they're going to play back and forth. Okay? ADH, aldosterone, in the renin-angiotensin system. The key here again is water follows sodium. Okay? So ADH, it binds to these principal cells of the lumen of the tubule, increases migration of vesicles containing aquaporins to the membrane. So if more aquaporins are going to the membrane, more channels to allow water through, so things get that you can increase water reabsorption. Okay? And then you end up with ADH, okay? There is this one right here. ADH. Some water reabsorption is independent of sodium. Okay? So water follows salt, and you can control urine production or water reabsorption by reabsorbing sodium, but then there's also a separate mechanism. Okay? So here's ADH. Here's a stimulus. Detect low blood volume, right? Or chemoreceptors within the hypothalamus. Increase blood osmolarity. Okay? meaning that it has a higher concentration of solutes, right? Lower concentration of water in the blood, right? So low blood volume, what do we want to do? We want to counteract that stimulus by reabsorbing water. ADH is released, okay, into the blood, and what does it do? It affects the hypothalamus, right? Increased water reabsorption, decreased water loss, right? And it works to counteract that stimulus. There's also vasoconstriction and activation of the thirst center. The thirst center. Because where do you get water in your body? What are the sources of water in your body? Don't look away from me, Abby. Like I'm not going to call on you if you'll go away. No, don't look at me. Stop looking at me, Swan. Where do you get water? What you consume, right? Now, most of it comes from what you consume every day, right? In the food you eat and the drinks you consume, okay? There's also some that metabolic water from the breakdown of proteins and carbohydrates. There's, right, water that is produced, okay? But most of it comes from there. And ideally, water in, how much you consume, should equal how much water you lose during the day. How much urine you produce, how much water vapor you breathe off, how much sweat is released, okay? Urine and all of that water loss, they should be equal in order to maintain hydration, okay? Now, some nutritionists will say you actually should be above that, right? You should have more water in than more water out, right? But that's a certain limit of that happening, okay? But these hormones, ANP, ADH, and aldosterone, along with the renin-angiotensin system, which starts this whole process, regulate water balance in the body, okay? And all of it is by regulating sodium reabsorption and water reabsorption down in the nephrons and in the kidney. Okay, so here's what it looks like in terms on the cellular level. Water moves through aquaporins based through osmosis. More aquaporins on the membrane, more water moves through. Okay, and ADH increases the number of aquaporins, eventually leading to an increase in blood volume. Right. Other materials that are regulated, potassium. Okay, potassium is regulated reabsorption. All right, most of it occurs in the proximal convoluted tubule, some of it in the reabsorption in the nephron loop. Right, this depends upon the movement of sodium. All right, so sodium and potassium are also linked. Why are they linked? What molecule or what carrier protein moves sodium in or out? What's important in the movement of sodium and potassium? The sodium-potassium pump, right? So it's logical that these are related. The sodium-potassium pump is going to move these in opposite directions. Okay, but they're dependent on each other. Okay, it also is dependent upon aldosterone. Okay, because aldosterone will move sodium, therefore it will also move potassium. Okay, the principal cells where sodium starts to be reabsorbed, these cells over here are responsive to aldosterone, and they start to secrete, get rid of potassium in order to keep the levels at a normal point. Okay? Other examples, calcium phosphate. All right, and this is regulated by a hormone, parathyroid hormone. All right, so calcium phosphate, and I don't want to go too much into this, I just wanted to let you know, 90 to 95% of calcium and phosphate is filtered as blood passes through the glomerular capillary. So that all has to be reabsorbed. Okay? Okay, so parathyroid hormone inhibits reabsorption of phosphate, right, and stimulates reabsorption of calcium. Parathyroid hormone is also important in what process of the body? Calcium and phosphate are important in what process of the body? What other system? Calcium important in what other? Skeletal system. Okay? So parathyroid hormone will help regulate the production and the deposition of calcium. Okay? So if you have low blood calcium levels, parathyroid hormone is released and stimulates reabsorption from the kidney. More then that brings the blood calcium back up to normal. You end up with regular. Okay, other examples, bicarbonate ions, hydrogen ions, and pH. Okay? This is important. These two, bicarbonate and hydrogen, okay, they help regulate blood pH through the buffer system that we discussed in blood, right? So CO2 is moved from waste cells or from the waste, the interstitial fluid, into the blood and is transported to the lungs in the form of bicarbonate ions, right? But if you don't have enough bicarbonate ions, you're getting rid of them. What happens to blood pH? It changes, okay? And you end up with too much and blood pH goes too low, okay? So this helps regulate this. We call it acid-base blood balance, okay? So if blood is acidic, what is acidic blood? pH of what? All right, 7, below 7.35, okay? So if it's acidic, the synthesized bicarbonate ions are reabsorbed into the blood, right? Within the cells. That helps to increase blood pH and decrease urine pH. Okay, so we're going to reabsorb bicarbonate ions, and at the same time, when I take up more bicarbonate ions, I'm actually getting rid of more hydrogen ions. So the urine that's being expelled will be acidic, below normal, right, below pH 6, but it will help to increase blood pH, because that's more important than whether or not I have acidic urine. Understand? Conversely, if blood is basic, okay, above 7.45, what do I want to do? Do I want to reabsorb bicarbonate ions? Or do I want, what ions do I want to reabsorb in order to bring the pH back down into the 7.4 range? Taylor, we're going to reabsorb hydrogen ions, okay? So here, if blood is alkaline, we're going to secrete the bicarbonate ions, get them eliminate them in the form of waste, but reabsorb hydrogen ions to bring the blood back down to the 7.4 ideal pH. Okay, so it's kind of like this balancing act. And we can regulate this based upon how much bicarbonate or hydrogen ions we're keeping in the urine. Okay, so you'll also see these two are inversely proportional, right? If blood pH goes down, urine pH goes up. If urine pH goes down, blood pH goes up, right? Because they're going to be opposing or opposite. Okay, about 80 to 90% of bicarbonate ions are reclaimed after filtration in the proximal convoluted tubule. The other 10 to 20% are reclaimed in the nephron loop, right? But here in the distal or in the collecting ducts, the reabsorption of bicarbonate and hydrogen ions in secretion in those collecting tubules, depending upon the blood pH that's present. Okay, so you can see this is kind of breaking it down. CO2, right, tubular fluid. Here's bicarbonate ions. This is the same thing we've had in the blood, where CO2 and you have bicarbonic acid, and it splits into CO2 and water. CO2 crosses the membrane, again goes into bicarbonate, and then splits hydrogen ions back out here, and the bicarbonate ion is moved to the blood. Okay? These things don't cross the membrane very easily. Okay, so we have to switch it over to CO2. That crosses the membrane easily. It reforms with water, and then we can split it into the bicarbonate ion and the hydrogen ion, which will be expelled back out to the body. Right? So we're splitting these 2 points. One bicarbonate to the blood, one hydrogen ion to the tubular fluid. What would happen if the pH of the blood was higher? If it was alkaline, this would be switched, right? The direction would be switched. We'd get rid of the bicarbonate ion and move the hydrogen ion to the blood. It's just more of direction. Okay? And that's what you see here. All right. Type A or type B cells is what directs where things go. Questions on tubular reabsorption of those materials? No? Yes? Maybe? Okay. So tubular reabsorption and the processes of the proximal convoluted tubule and the loop of Henle and the distal convoluted tubule, especially the loop of Henle, depend upon concentration gradients. Right? They depend upon changes in, or differences in the amount of materials in one area versus another. And then they're going to move things across, move things around. This is especially important in the medulla of the kidney. Okay? And the production of this concentration gradient is extremely important. All right? Extremely important in the reabsorption of sodium, extremely important in the reabsorption of water. Okay? And so there's a whole mechanism of how this concentration gradient is produced. Okay? And then we're talking about the interstitial fluid, the fluid between the blood and the tubule. Okay? And so the vasa recta, right, the blood vessel that surrounds the nephron loop, and the nephron loop are essential in making this happen. Okay? And the structure of the nephron loop is the first important part. Okay? We talked about channels and the production of channels and those molecules that are present to move things in and out. And if they're not there, things don't move. Right? More of them, more movement. Less of them, less movement. Well, the loop of Henle, the nephron loop, is important in this process, and it is because of the channels that are present in these 2 areas. Okay? We have a descending limb, different where it's going down, and then the ascending limb where it's coming out of the medulla towards the cortex. The 2 sides, the descending limb and the ascending limb, are selectively permeable to water and salt. Okay? The descending limb is freely permeable to water. Okay? Water moves across. But it will not allow, it has no channels to allow salt to pass from one side to another. Okay? So if the outside of that descending limb, the fluid outside of it, is high in salt, water's going to move across. Right? And as water moves out, what happens to the percentage of water inside the fluid within the tubule? Water's moving out. Water's moving out. And as it gets further down, water moves out. Okay? And now it gets to this point right here. Tubular fluid is more constant. It has 1,200 milliosmol. All right, you'll see this in the next. All right, so you see this right here. Right, 300 to 1,200 back to 300. Right, 300 to 1,200 back to 100. Because this side is impermeable to, the ascending limb is impermeable to salt, to water. The descending limb is impermeable to water. Right, so things move back and forth. back and forth. This is also important in terms of the blood. What happens? This is counter-current multiplier system. What's the two currents that we're talking about? Blood is one. What's the other one? Emma? The fluid, the tubular fluid. So counter-current means they're moving in opposite directions. And as you see here, if you can kind of look at the numbers, you can see that's about 300, 300, right? 300, about 100, but at the elbow, they're about 1200. So as you move further down the nephron loop, it becomes more and more concentrated. Okay? That allows stuff to move into the interstitial fluid and helps maintain that gradient, right? That gradient that is on the outside, right? Which will allow water to move out of here, out of the nephron loop and into the interstitial fluid. That makes it better, okay? So the two sides, let's actually go here, okay? We'll start at number 2. Water moves out, okay? Sodium on the outside of the nephron is pulling water out because sodium can't move back in, so that concentration gradient is always going to be produced. And as you move down the descending limb, you get more and more concentrated, less and less water, till you reach the elbow. All right? And at the elbow, it's about 1,200 milliosmoles. Okay? Now on the other side of the limb, well, guess what happens? The other side of the limb is not permeable to water, but is permeable to salt. So what happens now? Now you have this really, really high salt fluid that's at the elbow, and it starts going past these cells that are permeable to salt. But water can't move in. All right, water can't move in on this side. So salt starts going out. And as you go from the elbow back up towards the top, you are becoming less and less, right? That concentration is going back up. Okay, so we start, and this is a positive feedback mechanism. So here, in the thick ascending limb, salt moves out. Okay? Salt moves out and it produces a concentration gradient in the interstitial fluid. On the other side, water is leaving the cells, leaving the loop, because it is over here, right, permeable to water. So water moves out, tries to rectify that concentration gradient. This one to start, actually. Okay, so water moves out, makes it more even, and as fluid moves back through, right, sodium starts going back. It's all about countercurrent that's ongoing. So 1, 2, 3, 4, right? High things move out based upon the concentration gradient of the interstitial fluid around it and the tubular fluid inside. Right? This helps to concentrate urine. Helps to allow and maintain the concentration gradient. All right, so here's all the the numbers. Okay, so you go from 300 milliosmol eventually to 1,200, means really concentrated, less water, and then that's all because no salt can get out on this side, and water gets out based upon the differences in concentration between this and the vasa recta and the tube blood around it. Okay, so high salt, water's moving this way, right? But salt is moving out into the interstitial fluid. So these two countercurrents help maintain the concentration gradient of the interstitial fluid, right, which will help move things accordingly. Okay, last two things and we'll be done. Tubular secretion, right? We kind of touched on a bunch of them. Secreted substitutes include potassium, hydrogen ions, creatinine, right, which is a creatinine, not creatine, creatinine. Okay, they're related. Those two things are related, but this is a byproduct of muscle contraction. Creatine is used in muscle contraction. This is the byproduct of it. ammonia ions, some drugs, penicillin, etc. Okay? So, before you leave, hold on. This is the overview. Substances, where they're reabsorbed or they're secreted, and where they are. All right? Look at that. Make sure you understand it. And go from there. On Wednesday, we'll finish most of the urinary system.